Cancer, a deadly disease that terrifies individuals. With 9.6 million people dying from cancer annually, it is imperative that we create new treatments that are efficient and more accessible to the wider public. Cancer is caused by the rapid, uncontrolled division and abnormal growth of cells. The core reason behind this is due to DNA-level errors and mutations. The body constantly fights to eliminate precancerous cells. For instance, it was recently discovered that when cells undergo replication stress, this results in cell extrusion. Apart from this, mutations that occur in the oncogenes of DNA, coupled with the inability of tumor suppressor genes to function, can result in cancer. These cellular-level changes can be triggered by external factors, such as radiation and smoking, as well as genetic hereditary traits leading to increased susceptibility. Therefore, with cancer being a disease that so heavily impacts our society and individuals, scientists have tried and failed relentlessly to create a form of therapy to combat cancer. However, one form of immunotherapy seems to be immensely promising, and it may require us to turn to some of our deadliest foes, viruses. This therapy is known as Oncolytic Virotherapy. In its basic essence, oncolytic viruses are engineered to fight cancer by stimulating an immune reaction. Virotherapy is especially being sought after due to its favorability in accordance with the features of a cancer cell. This includes the ability of the cell to multiply and proliferate rapidly, the inability to undergo apoptosis, which is cellular suicide triggered either extrinsically or intrinsically, and the ability of the cancer cell to evade the immune system. This allows for the oncolytic viruses to have a wide reach within the tumor. The primary conventional method in which oncolytic virotherapy works is by stimulating immunogenic cell death. This is a form of cell death in which an immune response is triggered. This immunogenic cell death serves as a trigger for a cascade of patterns and the release of specialized cells. Firstly, the pathogen-associated molecular pattern, PAMPs, are activated. This activation is the beginning of the process. PAMPs activate the cytokines and the damage-associated molecular patterns, DAMPs. DAMPs and PAMPs help in serving as danger signals. Together, they help stimulate an inflammasome response. These inflammasomes are oligomers that are necessary for inflammation to begin. Along with the cytokines, they block the immunosuppression that is a result of the tumor. The cancer cells that are infected by the oncolytic viruses have different antigens on their surface. This difference in the virus-specific antigen allows for the identification and therefore destruction of the cancer cells. This final step is conducted by the T cells, which are immune cells designed to eradicate foreign particles and microbes. This, however, is only the more conventional method. There are a plethora of other methods that could be implemented to combat cancer. In fact, we may even be able to develop vaccines that are anti-cancer. For example, in one study, scientists created a vaccine with oncolytic viruses that also coupled with antigenic peptides instead of relying on the replication of viruses, which may be unpredictable. These vaccines may even be personalized for a specific tumor. In 2015, the first oncolytic virotherapy was approved by the FDA. This was the herpes simplex virus, designed to target melanomas. This is certainly an indication of the bright future that we have with oncolytic viruses. In retrospect, it seems almost impossible to use viruses, the harbingers of danger, to fight one of the deadliest diseases that we know. However, as Marie Curie once said, "Nothing in life is to be feared. It is only to be understood. Now is the time to understand more so that we may fear less." This is The Scientific Mind with Ishtanetra Siva.